Good afternoon, everybody. Thank you so much for joining us for today's Leaders Initiative seminar. My name is Stephanie, and I will be monitoring things here on the back end. Mark your calendars. Our next Leaders Initiative talk will be Monday, May 19th. We have Jin Wang will be presenting Uncovering Modifiable Environmental Drivers of Aging-Related Diseases, Insights from Real-World Studies. So that should be another really interesting talk. I will be sending out a Qualtrics evaluation towards the end of this, so please take your time and fill out the Qualtrics evaluation so we can address everybody's needs and wants and make sure we're scheduling talks that are interesting to a large variety of people. And with that, I will turn things over to Courtney for the introduction.
Great. Thanks so much, Stephanie, and thanks everyone for joining us today. We're excited to welcome Dr. Merci Best as our speaker for this month. Dr. Best is a postdoctoral research fellow in the Department of Neurology at the University of Michigan. Her research program focuses on Alzheimer's disease, progressive supranuclear palsy, and frontotemporal dementia. Her work aims to identify pathological hallmarks and genetic pathways as potential therapeutic targets for these neurodegenerative disorders. And with that, I'll turn it over to Dr. Best.
Thank you so much, Courtney. And thank you everyone for joining me on the Zoom. And I'm so grateful that you're going to record, Stephanie, so I can share the replay with others who weren't able to make it. So jumping in, I did want to give a little brief introduction to my scientific timeline. My focus for today's talk, obviously, is going to be on my postdoctoral work, which I started in 2023 with Henry Paulson. But it all goes back for me to 2008. Here's a cutout from a local newspaper, and I was able to do a research program at the age of 13, and I worked in a lab that studied Machado's Joseph disease. And funny thing, the professor of that lab is actually one of Hank's close collaborators and colleagues. So it all comes full circle with now me doing my postdoctoral work here at the University of Michigan. But in between that time, I've had numerous scientific experiences ranging from using zebrafish to now organoid models. And like I said, I'm super excited to share just a bit of that work today. So the title for my talk, as Courtney mentioned, is Unraveling the Complexities of Progressive Supranuclear Palsy and Frontotemporal Dementia. In this talk, I'm going to be going over two papers that I'm preparing for publication. Like I said, the first is going to be on progressive supranuclear palsy. And because that is a mouthful, I'm going to be using the acronym PSP for short. And the second paper is on frontotemporal dementia, where I will be using the acronym FTD for short. So my main research question that bridges these two seemingly different projects is, what are the shared molecular mechanisms between these two very different diseases? And my first line of work, which dates back to my PhD work at the University of Virginia in George Bloom's lab, focuses on the microtubule associated protein tau. So here is a schematic of the gene, the MAPT gene that encodes the tau protein, where you can see the different N-terminus inserts, the proline-rich region, and the microtubule binding region. And here is a schematic of a neuron where I'm segmenting the somatodendritic compartment in cyan, the axon initial segment in magenta, and the axon terminal in yellow. And I'm highlighting the monomeric form of tau, which is highly enriched in the axon under healthy conditions. However, in numerous neurodegenerative disorders, this process called tau mislocalization occurs about 10 to 20 years before someone starts to show any symptoms of cognitive decline. What is thought is that this tau protein that's normally highly enriched in the axon begins to abnormally accumulate and aggregate into higher order structures called oligomers and fibrils, and they centralize in the somatodendritic compartment. And this is one of the earliest signatures that a neuron is sick and en route to dying. So as I alluded to, here is a QR code for my PhD publication, which is found in the Journal of Alzheimer's Disease, where I found that damage to this magenta region, like I said, the axon initial segment, is one of the mechanisms that is promoting this tau accumulation. So if you scan this QR code, you will be linked to the journal and you can read the full publication. But as I bridged into my postdoctoral work, I tested the hypothesis that this is not specific to Alzheimer's disease, but perhaps I would see this similar damage in tau associated diseases. And I turned my eyes to progressive supranuclear palsy, which I'll be calling PSP. And the reason being is that PSP is more rare, but it is a pure tauopathy, meaning any of the pathology that we're seeing is usually attributed to tau, whereas in Alzheimer's, in my previous study, some of that pathology could be attributed to another protein called amyloid beta. So jumping into this story, I wanted to highlight the amazing team that has been helping with this project. As you can see, it's multidisciplinary, ranging from the Department of Neurology all the way to IT services here at the university. So a bit of background on PSP. As I said, it is rare. There's about 20,000 Americans currently living with the disease. And what we know from neuropathological diagnosis is that there is the presence of these tau tangles in the frontal lobe, subthalamic nucleus, the midbrain, and pons when you look at postmortem PSP brain tissue. And this accumulation of tau is associated with damage to these individuals' ability to communicate. We also see some issues with their eye movement. And even for a small subset of patients, we're actually seeing some cognitive impairment. And one of the most striking symptoms is a motor impairment, where these individuals oftentimes have trouble with balance and walking. So the research question when I'm thinking about this project is, firstly, is there tau pathology in the hippocampus? As you saw from my schematic, the hippocampus wasn't really mentioned when it comes to neuropathological diagnosis. And this leads back to my hypothesis that I will see this tau pathology in the hippocampus, and my even broader hypothesis that it's going to correlate with impairments in the axon initial segment and impairments in cognition. Unfortunately, I don't have all the data to share about the axon initial segment and cognition, but I do have a few teasers that I'll put in the talk, and hopefully in about a few months or so, I'll have the final data set on the axon initial segment structure and the ways in which cognition may be impaired in PSP. So thankfully, I was able to collaborate with the Michigan Brain Bank and the NIH Biobank to acquire all the tissue I needed to do some Western blot analysis to just see what are the total protein levels in postmortem brain samples. I have 12 control cases. These are individuals who were not diagnosed with any brain disorder at their time of death. And then I have 23 progressive supranuclear palsy individuals in this study. And one thing that I wanted to highlight is that some of the samples were purely PSP as a diagnosis, whereas the majority of the samples had other diagnoses like gliosis, atherosclerosis, and actually one of them had intermediate likelihood Alzheimer's disease. So I did want to be very transparent about the cases that we're using in this study. And I'm doing some preliminary data analysis, including the non-PSP tissue, which I just got from the NIH Biobank. But here's just a pilot Western blot. Like I said, I have 30 samples, but this is just 12 of them. And I'm highlighting C is the control, P is for pure PSP, and P plus is for PSP plus other diagnoses. And as you can see, there are some differences at the total tau level. There's also some differences at the phospho-tau level. And I'm really intrigued to start to quantify some of these differences in my axon initial segment protein of choice, TREM46, which is also a pre-axonal region marker. But I haven't begun this quantification just yet, but I'm on the edge of my seat and excited to see what we're going to see when we're comparing these three groups of cases. But for now, I do have some data just looking at the pure PSP versus the PSP plus other diagnoses. So once again, on the top panel, I'm showing the total tau. And as you can see, there's differences from sample to sample. In the middle panel, this is the phosphorylated tau. Once again, we're seeing those heterogeneous differences. And finally, on the bottom panel, this is the TREM46 axon initial segment marker. And so we took this data and we performed linear regression analysis to see if there was any correlation among these three proteins. And what we found was a positive correlation between total tau and the axon initial segment TREM46 protein level. And this correlation holds whether you aggregate all the samples together. So each dot is an individual case. In the left graph, and then on the right graph, I'm separating pure PSP plus, versus PSP plus other diagnoses, and we're still seeing that same positive correlation. So as the total tau levels go up, the TREM46 proteins levels go up as well. Next, to complement this Western blot analysis, where I'm taking this whole big piece of tissue, smashing it up, seeing what's inside, I wanted to do some immunofluorescence analysis to actually start to look at these individual tangles within neurons. And actually, began to see some tangles in glial cells as well. So once again, I did want to just show my cases. Here in this case, I do have 12 control and then 14 PSP. So unfortunately, not all of the cases that we had lysate for Western blot for, we didn't have the tissue to do the staining. But there were some commonalities among them. And
Merci,
yes.
Merci, Hank here. Elisa Torres asked a question which is relevant to this particular slide. So I thought instead of waiting till the end, I'd just bring it up now. She says, it appears as though those with PSP were all white. Is this typical in the general population? So I thought you might just want to know that she asked that question. Do you want to respond to that or not?
Oh, yes. Great question. And also, I cannot see the chat. So Hank, would you mind just unmuting?
I'll monitor the chat for you. I'll try not to interrupt you, okay?
Okay, thank you so much, Hank. So this is actually because of the sample that we have at the Michigan Brain Bank. Actually, there was one sample that was Asian, but the majority were white. In terms of the general population of PSP, I can get back to you on whether it's majority white or not. But I guess the main thing to know is that it's just rare in general. With this study in particular, we did only have white, but it would be interesting to see if we could get some racial diversity in the samples. Okay, so back to the immunofluorescence sample data, we also were able to see that some of these samples had other diagnoses, atherosclerosis again. In this case, we're actually seeing a sample that had age-related tau astrogliopathy. So it's going to be very interesting to see how the data pans out in terms of that correlation between the tau, because with the pure PSP samples, we'll be able to attribute that tau pathology because of the PSP diagnosis. But in some of these other cases, the tau pathology may be attributed to these other tau associated diseases. So here is a image of 3 individual cases. So in the left, I'm showing the non-PSP control brain. And what I did in this image was I blew the tau, phosphorylated tau stain up just to prove to myself that there's really no tau aggregates across this brain. So that's why you're seeing that yellow haze. In the middle panel is our an example of a pure PSP brain, where you're also beginning to see some of those tau aggregates accumulate throughout the hippocampus. And finally, on the right is the PSP plus other diagnosis. And this is one where you're seeing a robust amount of tau, and you're starting to see like tau that is not co-localizing with neurons in the blue cyan color. And so if we zoom into different subregions, you begin. Oh, Catherine? Yeah.
Yeah, could you go back for a second and just point to what I'm supposed to be looking at in the pure PSP compared to the other on the left and the right?
Yeah, so in the pure PSP, you're not seeing any of these yellow punctae, which you'll be able to see better when I go to the next slide.
Okay, great. Thank you. Yep, thanks so much.
Yeah, but in the pure PSP, there's like no punctae or aggregated tau the tangles, but you're seeing these little punctae in the pure PSP and the PSP plus other diagnosis. But here, this is where you can see it a lot better. So in this first panel is an example of none. So there are no phosphorylated tau tangles in this region of the brain. But in this next one, sparse, you're beginning to see a few of these little tangles, and you're actually starting to see these like neurophils as well. And then in CA4, you're starting to see a lot more, these nice flame-shaped tangles and some of these neurons. And then finally, in our robust category, it's pretty much every neuron has a tangle here. And you're starting to see even non-neuronal cells have tangles as well. So what we did was a semi-quantitative analysis to create this density map of tau phosphorylation. So in this, we're looking just at the PSP cases, and we're giving it a score of none if we didn't see any tangles, a score of sparse if we saw a few, moderate if we saw a little more, and robust if we saw a ton of tangles. And as you can see here, it is various between case to case. So in some regions, you're seeing a lot more robust tau pathology, and then in some regions, you're barely seeing anything at all. And so one thing I want to mention is that with this data in hand, we're now working on basically doing some machine learning approaches to take this semi-quantitative analysis to a fully quantitative analysis and actually count the number of tangles across these different brain regions, and also counting the axon initial segment number, but also doing some data analysis of the length and the intensity with the hopes of seeing if there's a correlation. My hypothesis is that in areas where you see high tau phosphorylation, there will be low axon initial segment numbers and shorter axon initial segment lengths and reduced intensity. And one other thing that we've begun doing is descriptive analysis. So with this, we have a sample size of 30, and we were not able to do a quantitative analysis to see if the cognitive scores correlate with the tau levels, but we hope to do a proof of concept study and request a larger data set of thousands of PSP samples and cases to see if patients, when they were living, have lower cognitive scores, but high tau levels when they donate their brains to research. So this is ongoing, but like I said, we have a sample of 30. We have a range of time of death. We have, once again, more PSP plus other diagnosis than pure PSP. And the MoCA scores do range. Some were about 2 years before time of death, and some we didn't even have information on. And like Elisa asked about, the race was only one Asian, and then the rest were white. And we had a good mix of male and female. So in summary, we did find that total tau and the axon initial segment do positively correlate in terms of the protein levels. We're also seeing heterogeneous phosphorylated tau in the hippocampus, And we think this is giving us rationale to move forward with requesting a larger data set to determine if we can see a correlation with cognitive decline. And also in the background, we're doing that machine learning approach to see some relationships with the axon initial segment structure. So with that, that's the end of the first part of my talk. And I did want to acknowledge the individuals who helped with this study. Again, I wanted to also open the floor for any additional questions. I know Elisa and Catherine had those questions, but are there any other questions on this PSP story before we pivot over to the FTD?
Merci, I'm just curious.
Yes.
When you quantify as few, moderate, like you said, you saw a few, how do you determine what's few, what's moderate, what's a lot? You just...
Yeah, so that it is semi-quantitative. So we basically looked across all 14 brains in this case. And it was actually 2 independent investigators, myself and Hank. We sat independently with the images and we created our own scoring system. But the limitation of that is it's semi-quantitative, and that's what's pushing us further to do the machine learning approach so we can get some actual numerics of there were 18 tangles in this region. And we think that will be a lot more elegant than our initial semi-quantitative approach. But it was 2 independent investigators and our scores were averaged across to get those numbers.
Right, it looks like
So
there is a-
related question, sorry, from Elisa, it's about the counting of the tau. Has this technique been shown to be valid, reliable? Is this something you test and publish? Let me just add that, a 4-point semi-quantitative scale has been used. It's not ideal, has been used in numerous neurodegenerative model systems, mostly by John Trojanowski and his team in the past. And I would agree with Merci, it'd be ideal to move this to a fully quantitative scale if you can. But a 4-point is about as far as you want to go in terms of going from none to robust. Merci, do you want to add to what I just said?
No, so I think you hit it right on the nail that this technique has been published in the literature. In fact, when you look at the literature, especially in the hippocampus, there's only a handful of papers and all of them have done semi-quantitative analysis. But I think the most rigorous approach would be to get some numbers there, like I said, with the actual numerics of saying, in this region of the hippocampus, there were 18 tangles, and then comparing that way.
Merci, can I ask a question?
Yes, you can.
You have your model is that there's going to be, if you will, a negative correlation between the presence of tau pathology and axon initial segments. And how do you square that with your Western blot data, which show a correlation that is kind of the opposite of that? I mean, I can imagine ways you could square it, but how do you think about that currently?
Yes, the Western blot data is meaningful, but it does have limitations. So when I'm running those studies, I have to take into account that I'm getting all the tau protein all over the brain. I'm getting all the TREM46 protein all over the brain. Whereas my immunofluorescence analysis is going to be more at the individual neuronal level. And I think that is going to give us more I guess more of a regional information, whereas I can say that the only TREM46 protein I'm assessing in the immunofluorescence analysis is going to be those that are in the axon initial segment, not elsewhere. Did I answer your question, Hank?
Yes, thank you.
Awesome. Stephanie, can you help me out? Any other questions? I can't see.
Yeah, another comment from Elisa. It might be interesting to compare your counting methods to results from machine learning.
Yes, absolutely, Elisa. I'm going to do that. And that will also help inform the field. And I hope that this machine learning approach can be adapted by others in the future. And it will be good to do like that compare and contrast to the semi-quantitative. Okay. All right. So the next story that I wanted to share is on the organoid model that I've been using to investigate frontotemporal dementia. And this is only going to be the study from the mass spec analysis that I did in collaboration with Irving Vega's group at Michigan State University. But I do have a handful of other sub-studies with these organoids that I'm still accumulating the data and doing the analysis for. So with FTD, we're actually seeing that there's about 50,000 Americans living with this disease. And here is actually a schematic I showed of tau, but I'm highlighting where this mutation V337M occurs in the tau protein. And this is one of the MAPT mutations that causes FTD. Now, I do want to orient you guys that there's only about 5 to 20% of FTD individuals living with FTD have it as associated with this with a mutation in tau. But this model system is going to give us a lot of information of how, in general, how we can start to find ways to combat or find targets that would be relevant for treating this disease. And so with FTD, we're seeing these tau tangles in the frontal and temporal lobes. And this, once again, does lead to some issues with individuals' ability to communicate. And then also their behaviors. We see a lot of unusual behaviors with those who have FTD. So to model this disease, I'm actually using cortical organoids from a protocol that was developed at the University of Michigan with Andrew Tidball, and actually seeing if we can use his methods to create these cortical organoids as a model of the human brain with FTD. But it's going to be very immature compared to the adult brain. So in this protocol, we start with induced pluripotent stem cells, and we go through a series of growth inhibitors and media changes to generate these 3D brain organoids. And for the process of my work, I collected these organoids every month for 6 months. And for the mass spectrometry analysis, I took 3 organoids at the 90-day or 3-month time point, and 3 at the 180-day or 6-month time point, and submitted them for mass spec analysis. So the research question guiding this project is, what is the effect of this mutation on cortical organoids' proteomic profile? And I hypothesized that I would see an upregulation of dementia-associated proteins when I received this data set. So here's a schematic. Well, this is actually an image from a wide field image where you can see this densely packed induced pluripotent stem cell layer. And then it's at day 3 of the protocol. And then by day 4, I'm able to cut this layer in small squares, and each square has the ability to fold on itself and form these organoids, which I see at day 9. And I pick these organoids out of a 3D matrix that they're in and plate them in 96 well plates until they're too big for the 96 well, and then I move them to 24 well plates. And here's the day 90 time point. In the left panel, this is the wild type organoid. In the middle is the heterozygous organoid. And on the right is the homozygous organoid. And now I'm showing the 180-day time point with the same order there. So when we submitted this, we actually identified 2,988 proteins. And here is the string pathway map of all of those hits. But I wanted to focus in, and here's a volcano plot of the 90-day time point where I'm comparing the wild type to the heterozygous organoids. So in blue are the downregulated proteins that were significant, and in red are the upregulated proteins that were significant. In gray is anything that was not significant, and in cyan is clusterin, which is the target that I'm moving forward with the validation process. So at 90 days, the clusterin protein was not significant, but this protein in the corner over here, annexin A11, was. So we've begun some pilot studies to assess whether this hit holds true, but we have been kind of still working on that and hope to have some more data soon. And then when you look at—
Sorry, Merci, can I ask a question quick?
Yes, you can. Yeah.
What do you think about this TTR signal?
Do I think it's real or not? Is that the question?
Yeah, like what do you think about it? I mean, yeah.
Yeah, so I did have the data specialist go back, and the signal actually did not hold true when he did the cleaning analysis of it. What he thinks is that it's just not present at all in the wild type, and then it's higher in the heterozygous. That's why it's coming up at such a high significance of upregulation.
Okay, because aren't there mutations in TTR that cause an aggregate like?
Absolutely. And so that was on one of our lists to follow up with, but it didn't really hold true.
Okay, great, thanks. Yeah, we see it in mice, and we don't know what to think about it, so that's helpful. I'll talk to you afterwards.
Yeah, I can give you the whole spreadsheet.
Thanks.
And then we also compared the wild type to the homozygous, and we're seeing similar trends of the upregulated and downregulated. A lot less proteins made this cutoff, and then the clusterin is here in the cyan. And so to the 180 day is where I'm focusing my validation on. That's where we begin to see clustering. It's kind of hiding out here, but it does reach our significant threshold in the heterozygous comparison. And then this actually holds true. It's up here at the top in our homozygous comparison. So we decided to move forward with validating clusterin to see if our mass spec findings would be validated by Western blot and also immunofluorescence analysis. So I did want to give a little background on clusterin. It's also known as apolipoprotein J. It is a glycoprotein that functions as an extracellular chaperone, and it's really instrumental because it prevents the aggregation of non-native proteins. But in the context of diseases, we see it involved in cell death, tumor progression, so lots of cancers, and also neurodegenerative disorders. And the main one is actually Alzheimer's disease, where it has been found in GWAS studies as a potential genetic correlate to the disease. But when I looked through the literature in frontotemporal dementia, I didn't see really anything about clusterin. So we're really excited to determine if this is a protein that we can validate and offer to the field as something novel and interesting to follow up on. So when I did my Western blot analysis on the left, you're seeing the wild type is the first panel, the heterozygous is the second, and the homozygous is the third. And then in the middle, I'm showing the total protein stain that I used to normalize the clusterin signal. And then on the right is how I graphed the data by the fold change, looking at the clusterin over total protein levels. And actually, we're seeing a statistically significant increase in clusterin protein levels in the heterozygous organoids, but that significance does not hold true in the homozygous organoids. And so we wanted to complement this Western blot analysis with some immunofluorescence imaging of the organoids. So in the first panel is the wild type, the middle is the heterozygous, and the right is the homozygous. And I just did a simple analysis of the max intensity here. And when we graph that, we're actually seeing an increase in the homozygous organoids that actually it's not statistically significant in the heterozygous organoids. But by eye, these are all projected at the same intensity level. So what we're seeing by eye does reflect the maximum intensity that is graphed. So that's interesting there. And so in summary, we did identify 2,988 proteins. What we did find in the data set was that when you compared the 3-month to the 6-month organoids, we're seeing a striking difference in the protein composition, which makes a lot of sense because as the organoids age and grow, we're starting to see more cell types involved, and that can lead to different changes in the protein composition. One thing to note is that we did observe tau in all samples and about 25 phosphorylation sites, but there were no statistically significant differences across those. And as I showed you some of the data, I'm beginning to validate this increase in clusterin. I'm seeing an increase in the Western blot heterozygous levels and an increase in the immunofluorescence homozygous levels. So it would be nice to start to do some more rigorous analysis to see what those differences are about, and can we even start to see what cell types we're seeing clusterin in? Is it in the neurons? Is it in the glial cells? And start to just do some more intricate studies with that data. And so with that, I'm showing the rest of the people that were involved in this research. I'm also highlighting here my mentoring committee, the different facilities and cores that I utilize to produce some of these studies. And I can't forget the funding resources through the university's T32 program, through the Michigan Pioneers Fellowship Program, and externally through the Alzheimer's Association Research Fellowship. So I do want to open the floors to questions on this part of the talk, and also even if you have questions on the previous part of the talk, I can address those now.
Hi, Merci. This is Taylor. Great talk. I was wondering if you have any hypotheses for that disconnect between the Western blot and the immunofluorescence. Yeah,
Yes.
that's really interesting.
Right. So Irving and I were talking a little bit about this. There are different ways these antibodies work based off of immunofluorescence and Western blot. So what we could be picking up on is a different distribution in the immunofluorescence, and that's why we're starting to see the patterns increase in the homozygous, but not the heterozygous. I also want to note that the analysis I did is not the most rigorous. So when I'm imaging these organoids, I'm taking 22 Z stacks. And for this analysis, all I did was pull the middle stack out and do like a just robust max intensity. But what I want to do is more at the individual level of seeing like different clusterin signals and normalize that to like a DAPI or a neuronal marker, and see if I see any more changes that way. But it could be, like I said, an antibody thing, just a localization of the protein change that we're picking up on. But it is interesting that we're not seeing. It is a trend. So I will say, if you look back at the, it seems to be like a trend of increase in the heterozygous and in the Western blot data. And then the trend is even more striking in the immunofluorescence. But I will definitely keep an eye out on that and start to think about like, why is there such a striking difference?
Yeah, that's really interesting. Thank you. I look forward to hearing.
I don't know if Elisa wants to ask her question that she's put in the chat or have
And
me
I
say it.
also saw Kevin had a question. Kevin, are you still there?
Yeah. Hey, Merci. Yeah, really good talk. Thank you.
Thank you.
Quick question. Have you guys thought about doing maybe pathway analysis for any of the sort of things that popped up? And what are your
Yes.
thoughts about that? You have some clues or some hints?
Yes, that is the next steps, pathway analysis to see if we're seeing, I definitely think like the neurodegenerative disease pathways will be activated. I'm really interested in like the neuronal structural pathways, and seeing if there's any changes in that. But I will have to shout out Mariana Sierra. She's excellent computational person in the lab, and she's already on it. She's excited about doing those pathway analysis.
Very cool. And so, what is your sort of takeaway? How do you feel about this as a model system? Like your general thoughts about it?
Yes, I will say the organoid model system is immature compared to when we're talking about human brain tissue. But the fact that we're identifying proteins that are implicated in neurodegenerative disease tells me that there's something to be gained from using these organoid models. And another thing is that I stopped at the 6-month time point, but we can take these models out longer. So it could be evidence to suggest that maybe try it. I mean, I personally don't want to do this, but a 9-month time point could be really elegant to see. And another idea that Irving had was to actually compare our mass spec data from the organoids with human data sets and start to see if we're seeing similarities, like are the hits that human tissue sets find with FTD, are they hits that we also found in our organoids? And that will just further increase our confidence that this model system is a mimic. It's not perfect, but it is a close mimic to what we're seeing in the human brain.
Okay, very cool. Thanks.
Thank you.
Mercy, Elisa did ask, he said, I did a quick search on PSP and race. It
Okay.
appears as though the majority of PSP is observed in whites. If this
Oh.
is the case, then maybe the control group should be all white as well.
Oh, well, so with human tissue, you have to really take what you can get, Elisa. So we were able to get 3 Black American of the controls, and we were super excited about that. So we will not be excluding them in this study, but it is interesting to know that PSP is majority white. And so our PSP sample does reflect that, but we definitely will not be excluding our Black American samples. I think they will add a lot of value to our study.
Well, I think you can do the thing, Mercy, where you ask if you leave those 3 out, do you still reach the same conclusion, right? Like, I think it's reasonable to be able to test whether or not you have a certain set of samples based on race that could be driving a signal.
Yeah, and I mean, 3 is not a lot when we're thinking about these studies, so I can play around with it, but my concern is reducing my sample size, which I don't want to do at all. But three shouldn't be a big deal. Yes, Irving, I see your hand up. Oh.
Yes, hi. Thinking about the PSP and the FTD, but mainly in the PSP, you saw a difference between what you call pure PSP and PSP plus other.
Mm-hmm.
The tau 5 and all your semi-quantitative data includes all PSPs, the pure and the pure plus other diagnosis, or how that the brains that you did for the semi-quantitative is the PSP only pure PSP or PSP plus the other?
It's both. And so with that, we just did a heat map showing all of them together, but the top 8 were pure, and then the remaining 6 were plus other diagnosis.
Okay. Those other I'm sorry if I might miss that, but that
okay.
What is that other diagnosis mean? What are those?
There were some that had atherosclerosis. There were some that had gliosis. It ranged. I can show you the table.
Okay, yeah.
But it was like a lot of other diagnoses. Some could be related to tau, and some probably aren't at all. I just wanted to be very transparent with the cases.
Good, because that's an interesting separation that maybe you can pursue. I know you have a very small case numbers and sample, but Similar to what Catherine said, if you segregate them, you see that your data is skewed by one group versus the other. That will be very interesting to follow too.
Absolutely. Yeah. I agree.
Just want to make a comment. I think that this goes back to Lisa's looking at the frequency of PSP in blacks versus whites. This is a disease that is often misdiagnosed or not diagnosed. And I wouldn't be surprised if, in fact, in different populations, it is underdiagnosed because it's missed. And I have to do some looking in the literature myself, at least, to look into that. But I wouldn't be surprised if there's an underdiagnosis of the disease that doesn't reflect the actual biological prevalence of the disease in Black people.
Absolutely. So if there aren't any other questions, I did want to kind of give you guys a vision for what I'm thinking about as I move towards writing independence grants and transition awards. Stephanie, were there any other questions in the chat that I'm missing? Or Hank?
No.
Nope, nothing there.
All right, thank you. So as I said, as I start to think about independence and like what I want to focus on in my lab, I'm really interested in this intersection between social and cellular factors and their influence on Alzheimer's disease risk. And so I wasn't able to talk about this today, but I worked with a team of researchers at the University of Michigan and at other universities to take a data set, the health and retirement study, and perform a secondary data analysis. So if you screenshot this QR code, you'll go to the link to our Med Archives paper, which has been changed slightly before we submitted it to the Journal of Alzheimer's. We're still waiting to get reviews back on it. But in this study, we took older adult population, and we wanted to assess the role of early educational experiences on disease risk. And what we found is that if you look exclusively at the older Black adults in this data set, their racial composition of the school did influence their cognitive health outcomes later in life. So as I began to think about how this could integrate with some of the cellular mechanism research I'm doing, I would love to find early risk factors that I could model in organoids. So for example, maybe we can't model racial differences of a school, but can we model stress that is related to these racial differences and start to integrate this secondary data that is out and publicly available with ways that we can find early risk factors and begin to start to target them to ensure that the Alzheimer's disease risk is minimized as possible. So the second wave of my work will definitely be looking at pathological assessments. So taking these postmortem tissue and doing Western blot and immunofluorescence that I've already carved a strong background in. And as Elisa alluded to, like starting to get more racially diverse tissue and starting to see if some of the pathological hallmarks that we think are instrumental to the disease, are they instrumental across populations? We're starting to see that play out in the Alzheimer's field with the amyloid beta drugs that oftentimes African Americans don't meet the criteria to be included in some of those trials because of the low amyloid levels. So starting to see what are the pathological hallmarks of the disease across a representative population. And then finally, moving from the data to the tissue to these organoid brain models and starting to do genetic manipulations that are maybe instrumental for risk for Alzheimer's that are in representative populations. So we already know that there are about 20 different genes that are coming out in African American families with Alzheimer's disease. So could we start to genetically manipulate some of those genes in these organoid models? And even further, can we start to use induced pluripotent stem cells from African Americans with varying levels of African ancestry and see, can we make some more representative brain models of the disease? So with that being said, I'd like to thank you all for attending my talk. I'd like to thank you also for the questions that you asked. I will definitely sit with this and integrate and continue researching and polishing off these projects. And also, like I said, thinking about transition fellowships and how I can start to carve my niche in the field. So thank you all. And if you have other questions that you didn't want to ask on the Zoom, feel free to send me an email and contact me that way.
Thank you, Merci.
Thanks,
Yeah, great job. Yeah, thanks so much.
Catherine.
Yeah, great progress.
Thank
Thank you,
you.
Merci. Bye-bye.
Irving,
Hope
thank
that will
you.
work.
Lots of positive feedback in the chat. Congratulations. Great job.
I appreciate it.
Yay. All right. Thank you so much, Merci, for doing this talk today.
Yeah,
And
thanks for having me.
yeah.
Great.
All right, everybody take care. Have a good rest of your day, and we'll see you at next month's Leaders Initiative talk.
Oh, Stephanie,
Yes.
are you able